Hello, everyone. It is my pleasure to invite you all, welcome you all to this seminar, another edition of Neuromath, where this time we have invited Dr. Carsen Stringer. She's recently started her own group in Janelia, and she's going to give us a talk on high-dimensional geometry of the visual cortex. A little bit of I hope everybody can hear me fine. I guess I should have started there, but um, I will try to give a little bit of introduction for her. So, Dr. Stringer did her undergraduate at the University of Pittsburgh, and later she did her graduate studies at the Gatsby unit in UCL under the mentorship of Dr. Kenneth Harris, where she worked on rodent visual cortex. And she's also been involved in the development of Suite 2P software. I'm sure many of you have come across it. It's very nice combined software that allows you to analyze 2-photon calcium imaging data. And after that, after her PhD, she started doing did a postdoc work at HHM Janelia campus, where she's now a group leader and has her own lab. Over to you, Carsen.
Great.
It's great to have you here.
Thanks for the introduction. So I'm going to be talking about high-dimensional geometry in visual cortex. I'll turn on my laser pointer so people can see it, hopefully.
Yep.
So great. So our brains are made up of 100 billion neurons, and these neurons form complex networks that both process external stimuli and drive the behaviors that we do. So to understand how the brain works, we need to really understand how these large populations of neurons work together. So we need to record many neurons simultaneously. So briefly, I'll tell you how we perform these large-scale recordings, and then I'll tell you about how we use these recordings to do science, in particular that we found that responses to visual stimuli in mouse visual cortex are high-dimensional. And then I'll tell you about a tool, a model that we've developed to try to capture the structure of high-dimensional activity in order to visualize neural activity. So how do we perform these large-scale recordings? We use a technique called calcium imaging. So this is the mouse brain. We make a small hole in the skull and cover it with glass. And so we can image through that glass day after day for months to a year at a time. And so what it looks like if you look through this little window is you'll see neurons firing. And when they light up, that means the neurons are firing here. And so often when people do calcium imaging, they're zoomed in on a subset of tens to hundreds of cells of field of view that's rather small. So but instead what we do is we zoom out and we record many planes simultaneously. So we record many planes simultaneously in depth, and this means that we have lower temporal resolution. We only have 3 samples per second, but this allows us to record many thousands of neurons simultaneously. And while we perform these recordings, the mouse has a screen in front of it, which is going to show natural images, and you'll see the images in the upper left here that are on the screen, and then you'll see each of these squares is one of these planes in depth of neural activity. And so there's still activity even when the visual stimuli aren't coming in, and then when the visual stimuli come in, you can see many neurons lighting up and responding to these stimuli. And now we capture these many hundreds of gigabytes of imaging data, and we convert it to a format that we can analyze using a tool which Pritish described called Suite 2P, where basically we find the neurons in these large-scale recordings automatically, and we draw these little circles. These different colors are different neurons. And then we sum the pixels in each of these neurons across time, and that gives us time traces for each of these neurons. So these are each of the traces down here. And so now we have these recordings, which are, in our case, we're thinking about neurons by visual stimuli. Now we're going to try to ask questions about the visual system. So I'll give you a brief introduction to the visual system and the question that I'm interested in, that we were interested in. So if you have the visual system, it starts with an image that comes into your retina, and the retina applies some post-processing filters, in a sense, to the image. And then the filtered image gets sent to the thalamus, and then from the thalamus to the visual cortex, there's this expansion of the signal. So there's at least 50 times more neurons in visual cortex than there are in thalamus. And there's two potential explanations for why this expansion might take place. So one possibility is that there's like 50 neurons, for instance, in visual cortex that are all driven by the same thalamic neuron, and they're all doing the same thing, all going up and down to the same stimuli. And this would constitute a low-dimensional code of the visual space. The advantage of a code like this would be that it would be robust to noise, that maybe a downstream decoder could have many neurons to choose from when trying to figure out what the stimulus is, but it's limited in the amount of information it has about the visual world. Another hypothesis would be that instead of many neurons representing the same thing, they're all representing different things. So each neuron represents a different visual feature. They're combining different thalamic neurons in nonlinear ways to create this large kind of filter bank of the natural world. And this might be advantageous for downstream decoders to be able to tell if this is a tiger or not, for instance, to have this high dimensional representation of the visual space. So we wanted to distinguish between these two hypotheses, and basically the difference between them is the dimensionality of the neural activity. So in this talk, we're going to try to answer the question, what is the dimensionality of neural activity? And to quantify dimensionality, we use a technique called principal components analysis and figure out what the eigenvalues are of the system. So let's say we have a matrix that's neurons by visual stimuli. We're going to take the principal components of this neural activity, and here's a diagram of this. For instance, let's say we take the first 3 rows of this matrix, and we plot them in this 3D space, where each dot here is a different stimulus presentation. It's a different column in this matrix. Now, if we take the principal components, what happens with principal components is the first one is a dimension along which the neural responses vary the most in the stimulus space. The second dimension is the second most amount of variation. Third dimension is the third most amount of variation. Kind of makes this, you can see this kind of box around the neural activity. And so the lengths of these axes are the variances, and these variances are the eigenvalues of the system. They're the variance of the neural activity, and then these are what we call the eigenvalues. And so we can think of let's say with this system, maybe we have 3 dimensions which have positive eigenvalues. And then the rest, if we took more and more principal components, PC 4, 5, 6, and so on, and they're all 0, then we get an eigenvalue spectrum that would look like this, where the first 3 are positive, and then the rest are 0. And this would correspond to a system where we can kind of put the system in a box that's 3-dimensional, and we can explain all the activity just in this 3-dimensional box. Alternatively, you could have a high-dimensional system where the spectrum is flat, and this would mean that every single variance is equal, and that the neural space is exploring many, many different dimensions. It's not able to be kind of contained in this box, this low-dimensional box. So let me make this a little more concrete with a couple examples. So let's say you have a low-dimensional system. So this is what the tuning curves would look like for an example of a low-dimensional system in response to a 1-dimensional stimulus. You have these wide tuning curves. Each color is a different neuron here. And this corresponds to a system that's only 2-dimensional. So you have 2 significant eigenvalues and the rest are 0. And if we look at a random projection of this space, then we get something that looks like this circle here. So what this means is that if I move a little bit in stimulus space, the representation is changing smoothly. It's not changing very much. And you can see that with these wide tuning curves. If I move a little bit in the stimulus space, one neuron kind of decreases its activity a little bit, the other one increases a little bit, and you kind of move smoothly through the space, through this stimulus space. And this is an example of a low-dimensional code. because you won't have quite as fine of resolution on the stimulus space, but you'll have this robust property. However, you could have something which is a high dimensional code where every neuron is coding for a different stimulus along this 1D vector. And so each of these spikes here is a different neuron. And this will correspond to an eigenspectrum that's flat. Each eigenvector is one of these neurons, and the variances are basically the magnitudes of the responses of these neurons. And if you try to move along this stimulus space, now you're going to have this problem where the representation is going to change very quickly. That's this spiky ball here. So one neuron turns off and a completely different neuron turns on. It turns off, a different neuron turns on. So you're not able to move. You're not able to move smoothly through the space. So if there's any kind of small perturbation, you're not going to know where in the space you're supposed to be, in a sense. So this is less robust, but you might have a finer scale resolution in terms of the stimulus that was being shown at any given time. So many studies have come to the conclusion that neural activity is low dimensional. But other studies have suggested that these studies have been limited in the sense that they didn't record enough neurons. So they're recording 10 to 50 neurons at a time. And so that automatically limits your dimensionality to at most 50. And also they're not showing many different stimuli at a time. So if you only show 5 different stimuli in your recordings, then you can have at most a dimensionality of 5. So in our study, we record many thousands of neurons, and we also show many thousands of stimuli, so that we have this really large matrix, and we can take the principal components of this matrix and say, okay, what is the dimensionality of this system, where we're not artificially bounded by the fact that we've recorded too few neurons or showed too few stimuli. So I'd like to just take the eigenvalues and be done, just take principal components analysis and be done. But we have we have a slight problem, which is that there's noise in the neural responses from trial to trial. So I'm going to show an illustration of this where we showed the same 32 stimuli to the mouse, and then we waited 5 minutes and we showed the same 32 stimuli again. And you'll see what happens in the video, that if there was no noise, you'd expect this video, the upper video and the bottom video, to look the same. So sometimes the neurons are firing at the same time, and you'll see some neurons that are reliable, but you'll also see a lot of differences between these two trials. So there's a lot of variability in the system, trial to trial, that we're worried about when we're taking the eigenvalues. I will just add, if you average over trials, you do get a reliable response. So if we look at the neurons, this is a neuron by stimulus matrix again, but now we're averaging over trials. So this is averaged over 48 trials. We look at this matrix versus if we average over the next 48 trials, and we see that they're very consistent. And there are also sparse responses. For instance, these two neurons seem to be sparsely activated to the stimulus. So there are tuned responses in visual cortex, and there are reliable responses if you average over many trials. But we're not going to be able to average over many trials because we want to show many stimuli. So we'll have to come up with a method to find the eigenvalue spectrum without the ability to average. All right. So how are we going to estimate eigenvalues from noisy data? So first, I'll show what would happen if I didn't take into account the noise. What would happen to my eigenvalue spectrum? So let's say I have this matrix that's neurons by visual stimuli, and we can define it as having signal phi plus some independent noise epsilon. Now what will happen, I'm going to do a simulation where I start with this pure signal and I start to add independent noise, and we're going to look at what happens to our eigenvalue spectrum. So the signal here, I've made this simulation have a signal that has this, that actually has this power law decay in the variance spectrum, and this is what it looks like without the log, not on a log-log plot. You have a signal that decays relatively quickly along dimensions. And so now if I start to add noise, what happens to my eigenvalue spectrum? And some of you might be able to guess, if I add Gaussian independent noise, what happens? I start to see that there's the eigenvalues at the end of the spectrum are going to be overestimated if I don't take into account this noise. So we need a way to remove this independent noise. We also need to be able to remove correlated noise as well across neurons. We know that there's correlated noise that exists in these recordings. We know that this is actually related to the behaviors that the mouse performs and is orthogonal to the signal, which is fortunate in this case. But so we need a technique to be able to remove these noise sources when we're trying to estimate the eigenvalues. So to do this, we use a cross-validated principal components analysis technique. And so we can't just do it with one trial. We need at least 2 trials of data. And so we show the same 2800 stimuli twice. And we use this second trial to be able to help cross-validate our eigenvalues. So I'll go through the way that this cross-validation works. So we have a matrix F, and we have, we're going to decompose it into its eigenvectors here, U, and then its right singular vectors here, V. And in this case, V has the singular values embedded into it. So U is orthonormal here. So we describe F as a sum of its eigenvector of this decomposition. And we're going to project U now onto not just F1, but also onto our second repeat F2. So we take these U vectors and project them into the first repeat of stimuli and into the second repeat of stimuli. This is going to give us 2 matrices, which will be number of components by number of stimuli. So each of these vectors A are each of these rows A are one of these vectors of U projected into the F space, into the neural space. And then what we can do is compare how similar are these projections from these 2 different repeats. And so we get an estimate of our eigenvalue sigma hat of K, which is going to be the dot product between this row and this row. And so we estimate, we have this estimation procedure, and in fact, what we show in the paper is that this is always going to be a lower bound on the eigenvalues, the sum of the eigenvalues that you have. So this is because the U that we find is based on just 1 repeat. So the U's that we find are going to have some noise in them that we can't get rid of because we've taken the U's on noisy data. So they're not just going to be the eigenvectors of the signal. They're going to have some eigenvectors of the noise too. So when we reproject these eigenvectors, which are not perfectly estimated into our data, they cannot possibly be the best eigenvectors. So we're always going to be below the estimate of the eigen. We're always going to have a lower bound on the estimate of the eigenvalues because the eigenvalues will always be the largest if you have the true eigenvectors, this sum here. The eigenvectors are the directions with the most variance, but we're not going to ever get to that point. So we're always going to be a bit below. That's why there's this lower bound.
Carsen, there's a quick question. Amo asks, what does orthogonal noise mean? I think he's referring to when you said noise is orthogonal to the signal.
Yeah, so that means that the directions so we have, you can think of we have the eigenvectors of the signal, and you can also estimate the eigenvectors of the noise. And in our case, we estimate the eigenvectors of what is noise based on the eigenvectors during activity without any stimuli at all. So we have periods in our recordings where we don't show any stimuli, and we estimate eigenvectors in those time periods. And if we look at the projections of those, we look at those projections, those 2 sets of eigenvectors, they're orthogonal to each other. So this is a really high dimensional space that's like 10,000 neurons. So the likelihood of even just like a random projection being different is not so surprising, if that makes sense. Like it's relatively likely that that a projection you find is going to be orthogonal to the projection, the true projection.
Thanks, Carson. I think that's the only question we have until now.
Okay. Cool. Yeah. So what that helps with is when we're taking these eigenvectors of f, which f has, like I was saying before, f has correlated noise in it. So when we take the eigenvectors, some of our eigenvectors will correspond to phi, and some of them will correspond to b. But because they're orthogonal to each other, when we project them into the neural space, we're only ever gonna get eigenvalues for b separate from eigenvalues of sigma or of phi, and those won't be consistent across the two repeats. So we won't have the eigenvalue, the b projections will not be the same across two repeats, because the likelihood, for instance, like the likelihood that the mouse is doing the exact same behavior and we have this exact same noise on these two repeats is very unlikely. So those eigenvalues, in a sense, will just go to zero if we have any eigenvalues that we find that are really related to this correlated noise rather than related to the signal. I don't know if that helped more or made it more complicated sounding. Sorry. Okay. So now we have this cross-validation technique where we're able to estimate where we have this lower bound on our eigenvalues. So now we have this technique where we're not just taking the PCs, we're able to get this lower bound estimate. So what does the eigenvalue spectrum actually look like? So if you recall from the beginning, we're kind of trying to decide if it's low dimensional or high dimensional, where low dimensional means that you have these wide tuning curves and only a few significant variances in your eigenvalue spectrum. Whereas a high dimensional system, you have this flat eigenspectrum where all the variances are equal. And what we found in the neural data was that it was actually neither of these. The variances actually decayed as a power law, where the nth dimension had variance 1 over n to the alpha, where alpha was close to 1. And so it's not low dimensional, because I can't just there's no cutoff where all these eigenvalues go to 0, all these variances go to 0. But it's also not high dimensional in this kind of sparse way where all the variances are equal. There's also these dimensions at the top of the eigenspectrum that have more variance, that kind of drive overall patterns of activity in the population to change. And so I'm going to show a little bit of validation for this cross validation technique with some simulations. And then we'll go into a little bit about why we think that this kind of is the optimal eigenvalue spectrum to have. So we did simulations where we added correlated noise in a space that was orthogonal to the stimulus space. And in this case, so the white is always the ground truth, and the different colors are the recovered eigenvalue spectrum from the noisy version of the simulation. And I should say, like for all of these, the signal variance is as a percentage is only around 15%. So we do have to add a considerable amount of noise to get to the level of noise that the neural activity has. So it's a relatively good sanity check in that sense, that there is a decent amount of noise in these simulations. So if we add correlated noise, we're able to recover a power law decay that's close to 1. If we add a multiplicative gain that's different on each trial, we're also able to recover the signal variance, or sorry, the signal eigenspectrum. If we add correlated noise and a gain, we're able to recover it, and also if we add the gain, correlated noise, and the independent noise, we're able to recover the eigenvalue spectrum and get alpha that's close to 1. So we did another validation. So this was validating the technique that we developed. And then we also did a validation where we wanted to see if we've kind of gotten to a convergence point in terms of our eigenspectrum being the true eigenspectrum if we had as many neurons as possible or as many stimuli as possible. So we aren't able to record more neurons, but we can subset our data and see what the eigenspectrum looks like at these different subsets, in these different subsets. So if we subset our neurons, we start with this blue and we converge towards this yellow. And you can see that this doesn't just look like noise, in a sense, that it looks like the eigenspectrum is getting closer and closer to this 1/n decay. And then similarly, if we subset stimuli, we get this convergence to the yellow again is all stimuli, but the purple starts above and they end up converging down. So if we look at the power law exponent, so this is 1/n to the alpha, we're looking at the alpha term, we see that as you increase neurons and you increase stimuli, they converge toward this value of 1 from above and below. So we think that we're in the limit of having recorded enough neurons and shown enough stimuli that we're kind of getting to the point where we're able to accurately estimate this eigenvalue spectrum up to a certain amount of dimensions, which will be, in a sense, limited by the number of neurons and stimuli that we show. Okay, so there's one potential reason for why we see this this eigenspectrum that decays as a power law, which is that the eigenspectrum of natural images has a power law decay too. So if you look at the eigenspectrum of natural images, this has been relatively well studied. It also has this 1/n decay where 1/n to the alpha decay where alpha is close to 1. And so it could be that the neural spectrum is just basically inherited from the image spectrum. So we thought, okay, well, let's change the image spectrum by, for instance, whitening the stimuli and see what happens to the eigenspectrum. And what we found in that case was that the eigenspectrum also decayed as a power law, where alpha is still close to 1. So it doesn't, even though we destroyed some of these low dimensions here correspond to slow spatial frequencies. Even though we've destroyed that low spatial frequency information, we still get this 1/n to the alpha decay where alpha is close to 1. And so, okay, we tried to destroy more correlations by basically only showing stimuli in a small region of the screen and removing everything else around it. We still saw a power law decay. And then if we destroyed even more correlations where we just basically showed squares of black and white on the screen and looked at the eigenspectrum, and we also saw that it had a power law decay. And so it doesn't seem like changing the image is changing the neural eigenspectrum. So we came up with kind of a hypothesis for what this kind of bound is on the eigenspectrum. Why is it decaying as 1/n to the alpha? Why didn't it get more flat, for instance, when we whitened images and so on? So we propose that there's this bound on the eigenvalues, so eigenvalue n, as n goes to infinity, such that the eigenvalue has to be less than n to the 1 plus 2/d, where d is the intrinsic dimensionality of the neural manifold, of the neural responses. So let me give you kind of how does the spectrum look like when the animal is in darkness? Oh yeah, so that actually does decay as a power law, but a bit faster, and I'll show that actually in a couple slides. Sorry, we got a question about what the eigenspectrum looks like when there's no visual stimuli, and it also does decay as a power law as well, but not with the alpha is a bit larger. Okay, so let me sketch this proof for you a little bit. So we're saying that we have our neural manifold has dimensionality d, and we're defining this dimensionality d is the box counting dimension. So what that means is if I say that d is 1, then I can cover my space with balls of diameter delta. And if I say, okay, if it's dimensionality 1, then I only need, the number of balls I need is going to be c over delta, where you can think of, so this one-dimensional line, you can think of it as having length c. So then I need at least c over delta balls to cover my 1D space here. So now what happens if I have a neural manifold that has dimensionality 2, then I have to cover it with 1 over d balls on this dimension and 1 over d balls on this dimension. So I need c over delta balls to the d, where d is 2. Oh yeah, sorry, I think I said d here. So I need 1 over delta balls on this dimension, 1 over delta balls on this dimension, so that becomes 1 over delta squared. And in this case, d is 2, because this is a 2-dimensional manifold. So this we can reformulate this. So this tells us if our neural manifold, it has a certain dimensionality, then we need this many balls to cover it. And so if we rewrite our equation in terms of the diameter delta, then our diameter delta has to be proportional to 1 over n to the 1 over d. And so in this case, the variance of the we care about the variance of the space because we care about the eigenvalue spectrum. So the variance is going to be the sum of the eigenvalues, and because we've kind of constrained our space to be within these balls, and the variance inside any given ball is at most going to be the diameter squared, because that's as far apart as the points can be, then you have the variance is going to be constrained by 1 over n to the 2 over d, because I've squared it now. And this is giving us a bound on the sum of the eigenvalues, because variance is the sum of all of our eigenvalues. And if we want it to be a bound on every single eigenvalue separately, it actually, you end up, if you do a little bit of algebra, you end up with this 1 here. So it ends up being that the eigenvalues themselves are bounded by n to the 1 plus 2 over d. So I got a question. I'm just going to presumably the power. Okay, so the question is that the eigenspectrum, I'm going to wait to answer this because I think the next slide will give some insight into that. So let me just go through, this is all very abstract. So let me show you an example of a system that has an eigenspectrum decay that is a power law. So let's say we have a bunch of cosine tuning curves. So each neuron now is a cosine with different frequencies, where n is the frequency, and also n determines how big the responses are. So the high frequency neurons have these smaller magnitudes, and this produces an eigenspectrum that has this power law decay where alpha equals 3. And so we've said if we have a manifold with this kind of intrinsic dimensionality, and this means that we have a continuous space. If we have a continuous space, then the eigenvalues must decay at least as fast as 1 to the n to the 1 plus 2 over d, where d is the intrinsic dimensionality. And in this case, our intrinsic dimensionality is 1 because we have a one-dimensional manifold. And so this means that n to the 1 plus 2 over 1 is n to the 3. So we want that where alpha equals 3 is where this bound is put, that it needs to at least decay as fast as this for it to actually be a manifold of dimensionality 1. If it decays any slower, it's going to end up being a fractal. It's going to not have an integer dimensionality. And so let me show a simulation of that. So first let me show you what this kind of being on the edge of not being a manifold and being a manifold looks like. So instead of that circle that you saw before where you move smoothly around, you have this circle that has kind of these divots and explores kind of some finer features of the stimulus space that you couldn't explore before in a low dimensional system. But let's see what happens as we change alpha. What happens to this kind of curve? So we're going to start at 4. So now we're well within the bound where the manifold should be smooth. And now we're going to decrease alpha, which means we're making this curve flatter and flatter. We're adding more and more high frequency components. And this is going to cause this to no longer be a manifold. It's going to end up becoming a fractal. So let's see what this video looks like. And so this kind of, you'll see, I can show it again, that this, I'll tell you when 3 happens here. So that's at that point where it no longer is this smooth manifold and it becomes this fractal shape. And so if you have more and more of these high frequency components and they have more and more variance, you're not able to explore your space smoothly any longer. So you want to be within this bound to be able to explore your space smoothly. And this is saying things about the neural manifold itself. We proposed that if the mapping from stimuli to neurons is smooth, then the dimensionality of the neural manifold itself must be at most the stimulus dimensionality. So there's this bound here. So we can think of D now as the dimensionality of the stimulus space. And if we think about that in terms of these high dimensional stimuli that we showed, we have this D is going to be high, maybe like 100 or something. Then we have n to the 1 plus 2 over 100. It's going to give us something like 1.02. So we kind of predict that alpha, in this case, this power law here, has to be greater than 1 for all of these different stimuli. So that's kind of the prediction that this mathematical proof gives, that if you think that the mapping is smooth, then you need to at least decay as fast as this. It doesn't prove that it's smooth. It just says that you need to be at least in this regime if you want to be smooth. And so this makes a prediction as we decrease the dimensionality. So now D maybe is around 100 or more. If we start decreasing the dimensionality, we show 8 for 1D, then we get different predictions for how fast the eigenvalue spectrum has to decay. So if D is 8, then we have 1 plus 2 over 8. And so we're going to get this 1.25 prediction. With 4D images, we're going to get 1.5. And for 1D images, we're going to get 3. So it's you're feeding into the system a lower dimensional object, like a lower dimensional space, and it hopefully what you want is that it's still going to be constrained to a lower dimensional space, because if it was exploring too many dimensions, you would no longer be exploring this 1D or 4D or 8D space in a smooth way. So if we look at our eigenvalue spectrum, indeed, when we change the dimensionality of the stimulus, not when we change these other properties, we see an increase in alpha. It becomes 1.5, and this is above the bound where we're thinking that the responses are smooth. And then if we show 4D images, this is also above this bound. It's alpha is 1.65. Then we show 1D images, we see that the power law also decays faster. So I'm going to answer some questions now. So there was one question that said that the power law decay in the eigenspectrum is still inherited from the power law of natural environments because the visual system has learned to expect to see the natural world. No. Basically, evolution and a life experience would lead to this power law, which is not stimulus dependent. Yeah, so I agree with that we know that the natural world, in a sense, is high dimensional. There's many different features of the natural world that we need to encode in our brains, and that suggests that it should be high dimensional. There's no reason why it would have to decay this in the same way as the image eigenspectrum. We're thinking that it's decaying this fast because it needs to be a smooth representation of the world. If you wanted to have as much information about the world, and actually these systems like efficient coding frameworks and so on, they actually don't have power spectrum decays. They have decays that are going to be more flat, and they're going to have more sparse responses. So it really depends on what kind of cost function you're trying to optimize. But we're saying that in this case, we're trying to optimize, in a sense, smoothness, that your responses to the natural world are smooth. And okay, is there another question?
Yes, there's a question in the Ask a Question which says, why is the true variance of the lower dimensions lower than the power law prediction, and that it only goes away when reducing the number of neurons, but not when reducing the number of stimulus dimensions?
Yeah, that's a good question. Yeah, so we think so. First off, I'll say the proof only applies to kind of the tail. in terms of the bound. So it doesn't necessarily predict that you would have to have a power law at the top of the eigenspectrum. We think that it might still be a power law at the top, but we're not able to well estimate those eigenvectors. So the problem with the eigenvectors at the top of the spectrum is we think that they're going to be corrupted by eigenvectors of the noise. So we think some of those eigenvectors will be eigenvectors of the spontaneous space. And in that case, we're going to be kind of underestimating them. And so I'm not sure why they would go away when you reduce the number of neurons, but not the number of stimuli. Let me think about that, and we can answer it later, because I don't want to go back to that slide now. That's okay. Yeah. Sorry. Okay. So I'm just gonna summarize this, and then maybe we can go back to that question. So we found that this alpha, the power law exponent, so how quickly the eigenspectrum decayed, depended on the dimensionality of the stimuli. And so we have basically this bound, which is 1 plus 2 over d, where d is the stimulus dimensionality. So I'm plotting 1 plus 2 over d on the x-axis here. And you can see that the alphas are all above this line, suggesting that they're all within the regime where the responses are smooth. And so we think that neural codes are balancing kind of capacity. They want to maximize the amount of information they have of the visual world. And so that's why this eigenspectrum is high dimensional. It's not just a low dimensional spectrum with only a few significant eigenvalues. It doesn't drop to 0. But it's also trying to optimize smoothness. So it's not just to be as high, if it wanted to be as high dimensional as possible and sparse, then you would have this flat eigenspectrum. But what we see instead is that it decays at this power law, so that it's still in a regime where it could have smooth responses to stimuli. You want similar stimuli to be represented similarly. You don't want to have this jumpy kind of representation of the world. So yeah, actually, yeah, I'll take some questions here before I move on. So I got one question which said that, how do you think this would look for another stimulus modality like auditory? We don't know. It could also be high dimensional in this way with a power law decay, but it hasn't. I don't think it's been tested before. So I think in general we think that it's a good idea to have kind of these low PCs which represent these overall changes in the contrast and other broad features of the image, and then these finer features which encode more fine scale properties of the images. Yeah, sorry, Pritish, do you want to...
Yeah, so I had a question at this point. Do you think the smoothness property says something about, because in the real world we don't really process images, we process videos, and videos are like real life is smooth, and is that the reason why the system is optimizing this, or would you make more specific predictions of if we showed video stimuli to the animals of something that we would see of how they move within the manifold or something like that?
Yeah, that's a great question. So first off, I'll say there's two aspects of smoothness you could have. So smoothness in the case you're saying is like an object moves smoothly through the world. There's also an aspect of smoothness which is like which can be category specific or object recognition kind of specific. Like you see a texture of leaves, and that kind of that changes in some small ways, and that gives you this category of leaves, and you want all those categories, the same category, to be represented similarly. So I think both of those are cases where you would want a smooth representation. You would want to smoothly represent both of those cases. And I think, and that's some of the work actually we're exploring now, is kind of changing changing the stimulus space in these kind of local perturbation ways, and seeing how the neural manifold changes with respect to those. And I think it's a great direction in terms of research to try to figure out what directions kind of keep you on the neural manifold and are represented smoothly. But we don't actually know the exact answer to that question.
Yeah. Thanks. I think there's one more question which asks, how do you think this power law would look for higher order areas?
Yeah, so we've looked in V2 and we see the same power law, but we've just looked in kind of what's defined as V2 for mice. But I don't know what would happen in higher areas, for instance, in monkeys. I think once you get to areas with object like IT and so on, you might not necessarily expect this power law. But like these, if you're in this, you're really far along in this category space, but it's totally open to be explored. Oh yeah, and we got another question. Is it a problem that PCA finds linear instead of nonlinear combinations of the input variables? It's a problem in the sense that it's hard for us to interpret what the manifold is underlying the neural activity. So we don't know if, from this linear analysis, we don't know if there's a nonlinear manifold underlying the neural activity that's a bit lower dimensional. We think that there might be something like the nonlinear space might be a bit lower dimensional than the linear space, but we can't. It's hard to interpret kind of the eigenvectors of the linear decomposition. But then again, there's also not great ways to explore kind of a nonlinear reduction of the space. And actually, that's one of the models that we've been working on is kind of trying to capture this high dimensional structure to try to understand what this nonlinear manifold might be. And I think related to that, like doing these local perturbation studies and so on are hopefully going to help us try to find this underlying manifold. It's a good question. Would this analysis depend on interactions between neurons? In other words, would you expect the same power law if neurons from which you record responses were isolated from each other? Yeah, so so this is always done on the covariance matrix. So this is looking at the correlations in the stimulus space, ideally, of the neurons. So these low dimensions are dimensions where the neurons are varying together. But in theory, you could record all the neurons separately, and as long as you're able to average their responses over trials, you could concatenate them and look at that eigenvalue spectrum. So you would still need it at least to where we are trying to estimate the signal. So any kind of noise that's outside the signal space, we're trying to ignore. So it would be okay if you recorded them separately. But the eigenspectrum itself is this way because the neurons are correlated to each other. If that makes sense. Yeah, so it's pretty late. So I don't want to. I'll just say that we have this kind of model that we've built called Rastermap that tries to visualize high dimensional neural activity. And I'll just show once. I'm not going to go into how it works, but basically it's a method that you have a neuron by stimuli, neurons by time, and it resorts this axis so that neurons that are correlated to each other get put close to each other in this one-dimensional manifold. So it's a way to non-linearly embed your neurons to try to see the structure. And so this is one of the ways that we're trying to explore this high-dimensional structure, which is difficult to understand, especially because a lot of the methods for nonlinear manifold analysis also are not super robust to noise, and we also have a lot of noise in our recordings. And so you should check out Rastermap if you're interested. I'm not going to go through all of this. This is what the stimulus responses look like embedded in Rastermap. Here's some zebrafish activity with Rastermap. And I'm just going to conclude. We found that neural activity is high dimensional, and that we think it's kind of doing this balancing act between being as high dimensional as possible, having as much information about the stimulus as possible, while still representing the stimulus space in a smooth way. And then also you can play around with Rastermap if you want. And you can circle, this is a two-dimensional embedding in this case, and you can circle clusters and look at what the neural activity looks like. And I'd like to thank my advisors, Marius Kakitsariou and Kenneth Harris. This was really joint work with both of them. And I'd like to thank all my collaborators at UCL. This was also, Kenneth and Matteo have a joint lab there, and it was great working with them. and Nick Steinmetz and Charo as well, and then my collaborators at Janelia as well. So that's it, and I'm looking for postdocs and students who are interested in coming to Janelia. If you're interested in working with me, let me know.
Okay, great. Thanks a lot for this talk, Carsen. Maybe we can wait a couple of more minutes to see if there are any more questions, since we have a few more minutes. To start out about Rastermap, so is there a reason why does it really help us a lot to just sort the neurons, or is there something more that you can do beyond just arranging them in a different way that allows us to see, I don't know, like more dimensions simultaneously? Because presumably if there are really if it's really 2D representation, then we're sort of projecting it onto the one dimension by sorting the neurons by correlation, right?
Yeah, well, it's not just a sorting in our case. It's not just like t-SNE in that sense. It's actually a model where we're basically, it's more of a matrix decomposition algorithm with this kind of nonlinear layer on top of it. So it does give us some you can do it in 1D, you can do it in 2D, you can do it in ND, and you kind of have this underlying model of the space that you can start to explore. So you can look at groups of neurons that are close to each other. When you sort it just by 1D, you can't tell that there might be separations between clusters. It's just a nice way to look at things. But yeah, so it's one way. I think we're going to need to develop new ways as well, though, to visualize these things and understand them. This is you're definitely throwing away information when you use a method like this.
Okay. Great.
Yeah, Rastermap if you use Rastermap in Sweet2P, it just basically resorts it in 1D, or if you run it, and you can just basically you can do this with any manifold embedding algorithm. You can run t-SNE with one dimension and just resort your neurons based on the sorting that t-SNE gives you. And what we do is we resort and then we smooth a bit, and that helps us to try to start seeing the structure.
Just Thompson asked a question. Does the potential computational role of correlated noise fit into the theories into this theory somehow? How would correlated noise affect smoothness of manifolds?
Yeah, so that question is kind of assuming that correlated noise is good. And so there's quite a bit of work by Peter Latham and others that are suggesting really correlated noise, especially in certain spaces, can actually be detrimental for coding. We don't actually see this detrimental noise in our recordings. We think that most of the correlated noise we see we're able to just kind of get rid of in the space, and so we don't think it's hurting us. I don't also think of it as necessarily helping us. If you think about it, especially in primary visual cortex, you want to have a representation of the world that's reliable and useful for you to use. So I think any correlated noise that's in the stimulus space is not necessarily a good thing. We think of the correlated noise. I'm sorry, this question is like huge. There's correlated noise that we see that's related to the behaviors of the mouse, and that's orthogonal. So it doesn't hurt stimulus coding. You can think of them in a sense as two separate manifolds. One that's driving the one that's driven by behavior and one that's driven by stimuli. And if we train the mice, maybe there will be overlaps between these manifolds, and maybe there will be shared features. But in the case where the mouse is just seeing stimuli and just running or whisking, we see that they're kind of orthogonal. And I think of that as probably a good thing, that the mouse can still see what's going on.
Right. Okay, I think we can stop here. Thanks a lot, Carsen, for accepting this invitation and this wonderful talk. It's also, just for everybody, it's also live streamed to YouTube. So I posted a link there, so you can find it there if you want to refer to it again. Thanks a lot, and see you on next time.
Thanks for inviting me. Thanks everyone for coming.